Welcome to the Introduction to OPAL Multiplex Immunohistochemistry workshop. This is being brought to you by the Center for Advanced Histology and Microscopy at the Peter MacCallum Cancer Center. If you want to get in touch with us after the workshop, then our email is cahm@petermac.org, or you can also follow us on Twitter at campmcc. Before we get started on the workshop, I just wanted to give you a brief introduction into our center. The center is made up of 4 complementary platforms, and these are histology, optical microscopy, electron microscopy, and image analysis. The center is staffed by 8 team members, myself, Paul. We also have Danya, Basia, and Leah, who take care of our histology platform. Judy and Alicia, who take care of the electron microscopy platform. Rayhan, who you'll be hearing from later on in the workshop, who looks after our immunohistochemistry core. And Meta, who you'll also be hearing from later on in the workshop, who looks after our optical microscopy and image analysis core. So why are we running a workshop on OPAL? So we have a lot of experience working with OPAL multiplex immunohistochemistry. We've had the Vector 3 system since 2017, and we also had the world's first BondRx system in 2018. And later on this year, we're expecting a POLARIS system to enable us to do 9-channel multiplex. Up to this point, we've had over 130 antibodies optimized for panels, over 1,500 imaging sessions in the last year, and we've developed panels not just for clients within Peter Mac, but also within institutions across Victoria and the rest of Australia. So this is just a quick table just showing you some of the 130 antibodies which have been optimized thus far. Today's workshop contents, so I will be giving you the introduction to multiplex immunohistochemistry, and I'll also touch a little bit on the immunostaining protocol, image acquisition, and data analysis. But Rayhan will also cover more in depth key aspects of the immunostaining protocol and image acquisition, and Meta will follow on and take you through our workflow for data analysis. Okay, so let's get started with a basic introduction into the background to OPAL multiplex immunohistochemistry. So, why do we want to use multiplex immunohistochemistry? So this is a schematic of the tumor microenvironment, which is extremely complicated with lots of different cell types within it. And what we'd really like to know is what's the spatial context between these different types of cells, which ones interact, which ones are more removed from each other. And this is very difficult to do using standard immunohistochemistry methods. So with standard immunohistochemistry methods, we're limited by the antibodies that we can use. So if we use something called direct immunofluorescence, and I'll come into this in a few coming slides, that doesn't give us any signal amplification steps. If we use an indirect immunofluorescence assay, then we can be limited by the number of antibodies or the number of species that the antibodies have been raised in. Similarly, if we use standard fluorescence microscopes, we're limited to about 4 to 5 channels based on the filter cubes which are available within those instruments. So you could do multiplex immunohistochemistry, but you'd probably have to use serial sections in order to do that type of experiment. What OPAL multiplex immunohistochemistry gives us or allows us to do is multiple stains on the same section and also multiple stains from the same host. So if we've got lots of antibodies that are raised in rabbit, we can still use those. Here at CAM, we have, as I mentioned before, the BondRx, which is used for automating that staining. And we also have the Vectra, and soon hopefully the Polaris, where we can image up to 9 channels at one time on a single section. Nice thing on the vector as well is we can preload in 200 slides and leave the system to run automatically and image all of those samples. We also have both Inform and also Halo licenses, and we have 8 Halo licenses available to our users to analyze the data that's coming off the microscopes. So, what is OPAL? So, if you're here and you're thinking that OPAL is a travel card which is used in New South Wales, or a rare type of gemstone which displays this iridescence pattern, then this probably isn't the workshop for you. If you're here because you think it's a method for performing multiplex immunohistochemistry, then hopefully the next hour and a half will be a little bit more interesting to you. So how does the immunostaining protocol work? So what we're going to go through is a little bit of background here into immunohistochemistry, and we're going to start off with a chromogenic stain, one that we'll talk about quite a lot within the workshop, which is one called DAB. In this case, we've got our tissue section here, and this little blob here is our protein that we're interested in, or our antigen. In this case, we've got an antibody here, which is detecting this antigen, and this is what we call our primary antibody. In this case, again, we've got a secondary antibody in here, which is detecting this primary antibody, and this secondary antibody has attached to it a horseradish peroxidase enzyme. And this enzyme can catalyze this reaction. It requires hydrogen peroxide, which takes the DAB from a non-colored state into a colored state, which is then deposited down onto our tissue. And this results in this brown staining pattern. Here we're using an anti-CD3 antibody in tonsil, and you can see where your protein of interest lies within the tissue. And here we've counterstained the nuclei. So this is our chromogenic stain using DAB. We can also use antibodies and use fluorescence to detect your protein of interest. So in this case, we've got our direct immunofluorescence assay where we have just a primary antibody. This is conjugated directly with a fluorophore, and we'll discuss fluorophores in the next few slides. And we also have the indirect immunofluorescence assay where again we have a primary and a secondary antibody that detects that primary, and it's the secondary which has the fluorophore attached. As I mentioned before, the direct immunofluorescence assay can be very clean, but doesn't give you that amplification step that you get in the indirect immunofluorescence assay, because you can have multiple secondary antibodies bound to this primary antibody. And this is the resulting fluorescence image taken on a microscope. Okay, so we mentioned and we've talked a little about fluorophores and fluorescence, so I thought I should take you through just in case you haven't heard of these before. So on the left of this slide, we've got lots and lots of different what we call fluorophores, which are basically any molecule which can undergo fluorescence. Now, what does fluorescence mean? On the right-hand side of this slide, we have what we call a Jablonski diagram, and what we're looking at is the energy state of the electrons within all of these molecules. So what's happening with these molecules is they're absorbing the energy from light. So a photon of light is being absorbed into these molecules. This is causing an excitation of the electrons within the molecules up to this higher energy state. Some of this energy is then lost, and normally this is lost as heat, down to a lowest excited state. And then finally, the electrons come back down to the ground state in what we call the emission step, which results in the release of a photon, which has a slightly longer wavelength, lower energy, because we've lost some of the excitation intensity here. So in this case, we're putting in a photon with a wavelength in the blue wavelength, losing a bit of energy, and that's resulting in emission of a photon in the green wavelengths. And we can use this within the microscopes to detect the signal. What we do have, so I mentioned there's two steps there. There's excitation and emission. And this can happen across a wide range of the electromagnetic spectrum. So each fluorophore will have very specific spectra. So here in the dashed line, you can see this is the excitation curve. This would be the same as the absorbance spectra for this fluorophore. And you can see maximum excitations happening at about 494 nanometers here. The jump between the peak of the excitation and the emission is that heat that's lost. So this is what we call the Stokes shift in fluorescence. And our emission spectra here in the bold line shows us the wavelengths over which photons can be emitted, again with a maximum at the peak of this curve at 525 nanometers. Now, depending on the structure of these fluorophores, these excitation and emission spectra will change. Okay, one last immunohistochemistry method that we'll talk about is taramide signal amplification. And the reason we're going to talk about this is because this is what we use within OPAL. So in this case, it's kind of a mixture between the colorimetric staining and also our fluorescence staining. So again, we've got a primary antibody here, which is detecting our protein of interest. And we have our secondary antibody, which is detecting the primary antibody. But in this case, our secondary antibody again has this horseradish peroxidase enzyme linked to it, like we had in the DAB staining. But the reaction which is happening is slightly different. So in this case, it's taking an inactive tyramide fluorophore and using hydrogen peroxide is creating a reactive tyramide fluorophore. Now what this means is that this fluorophore will then covalently bind to tyrosine residues in the vicinity of this reaction. So this means it will go on the antibodies, but it will also go onto the tissue as well. And this is really important, and we'll come to this in the next couple of slides as to why that's important. Okay, so how does this work in terms of how can we use it to do multiplex staining using OPAL? So as I mentioned, this is the same reaction step that we covered in the last slide. So creating the reactive tyramide fluorophores, because this is an enzyme, this can be done multiple times, which gives you signal amplification. And this is an oversimplification, but what essentially will happen is you'll end up with covalently attached fluorophores to your tissue. We can then do a heat treatment, which removes the antibodies from the tissue, but the fluorophore will remain. We can then come back in with another antibody, detecting another protein, do the same reaction but with a slightly different fluorophore, and this again will result in that fluorophore being bound to our tissue. And we can do this multiple times and build up our multiplex immunohistochemistry. This is the OPAL staining protocol for FFPE tissue sections. First, we'll deparaffinize by baking at 60 degrees for 1 hour, washing 3 times in xylene, and then rehydrating via an ethanol gradient back into water. We'll fix the tissue using 10% neutral buffered formalin for 20 minutes, followed by a wash. We will then perform antigen retrieval using either a pH 6 or a pH 9 solution with microwave treatment. And then we go through the normal cycles of immunohistochemistry staining. So blocking with a high protein solution, incubation with primary antibody followed by rinses, incubation with secondary antibody, again followed by rinses, and then our OPAL reagent reaction, again followed by rinses. We'll then go back through a heat treatment for antigen retrieval, And then we'll go back through this cycle, back to blocking. And we'll repeat this cycle for as many OPAL fluorophores as we have within our panel. And then finally, we'll stain the nuclei within our tissue using spectral DAPI before mounting the sample with a cover slip and imaging on the microscope. And Rayhan will go through a number of these steps in her talk in a little bit more detail. In terms of the staining kits which are available, so you can buy 4-plex, 7-plex Vectra, 7-plex Polaris, and 9-plex Polaris, and this changes the different fluorophore combinations that you have within your imaging protocol. Now, the 4-plex OPAL fluorophore you could use on a standard microscope using standard bandpass filters, and again we'll cover that in a few slides time. So we've got our DAPI, which is a peak at 460, OPAL 520, OPAL 570, and OPAL 690. But as we start going up to the 6-plex and then finally up to the 7-plex, you can see that it really starts to get complicated. You've got a lot of crosstalk and bleed through between each of your fluorophores, which really complicates the analysis. One other really important thing is when you get to the 9-plex or any of the Polaris stains, you'll notice that we're using OPAL 780, and the staining protocol for this is slightly different to what I've shown you before. So I'll show you this in the next slide. Okay, so for OPAL Polaris 780, again, we're following a fairly similar pattern, but we're doing it over 2 separate steps. So we've got our protein of interest, our primary antibody, our secondary antibody with our horseradish peroxidase. And in this case, rather than attaching our OPAL fluorophore, we're actually attaching a protein called DIG. So in this case, we're getting the same amplification, covalent attachment to the tissue, and removal of those antibodies. And then we can come back in with some more antibodies, and in this case we're going to use the OPAL 780 fluorophore, and because we've got so many molecules here, we can get lots of these antibodies binding. Again, that results in more amplification, which again will be attached to our tissue section before the antibodies are taken away. Okay, so in terms of our OPAL considerations, and again, Rayhan will cover this quite a bit in her talk, things we have to bear in mind are our single antibody optimization, and we're going to use that using DAB, as we've discussed earlier on how that works. Key things are things like our antibody dilution and our antigen retrieval. We'll then go through our monoplex optimization. So where should that stain sit within the staining order? Things like our antibody and OPAL fluorophore combinations. We will develop a library slide, so single stained and also autofluorescence controls, which are really important to getting good data out at the end of your experiment before going through your multiplex optimization to assess the balance, any interference and crosstalk between your fluorophores. And if we have to, we'll go back through and reorder where we're putting the fluorophores within the stain. Next thing we're going to look at is our image acquisition. So how do we separate out these stains? You saw with that 9-color, 9-plex panel that the fluorophores were really overlapping. So what we want to do is take this raw data and convert it into something where we're a little bit more sure where each of the fluorophores and each of the signals are coming from. So on a standard microscope, as I mentioned, you're pretty much set for 4 or 5 fluorescent stains, and this is because we use something called a bandpass filter. So for our DAPI channel, we're going to collect the wavelengths that are emitted only across these wavelengths. For our green channel here, only across these wavelengths. For our yellow wavelength across here, and then for our red wavelengths, only across these bandpass. And you can see there's fairly minimal crosstalk or bleed through between those fluorophores. What we're doing with the vector or the Polaris imaging is we're making use of what's called a liquid crystal tunable filter. So what this allows us to do is essentially take an image over a very narrow wavelength region, and this has down to a 20 nanometer resolution, and we're able to take not just an image in XY, as you would see here, so this is our YX image, but we can also take that across the wavelengths as well, across lambda. The vector and Polaris systems also make use of long pass filters and also some band pass filters as well. To do this, we require control. So we need individual opal stains within the tissue to basically say this is what the spectra looks like for each of the fluorophores. And we also need a reference for the tissue autofluorescence as well. So we're building up an image, not just in XY, but across the wavelengths as well. So this is the setup for the Vectra. These are the long pass filters, so it collects all of the photons that are emitted. So this is our DAPI filter from here along. And what we'll do is use the liquid crystal tunable filter to basically take images in steps of 20 nanometers across and build up that spectra that we can use to unmix the data. And we'll do that for each of the filters for each of the fluorophores. Some of the imaging considerations, and again Rayhan will cover this in her presentation, as I mentioned, is building that spectral library from single stain control slides for each of the fluorophores, for DAPI and for your autofluorescence, and to extract each of the fluorophore spectrum using Inform on the acquisition computer. We should try and aim to set our exposure settings within the 5 to 200 millisecond range. We will first of all initially image the whole slide and then select the regions of interest that we want to go on and image in our multispectral imaging or MSI regions at higher magnification. And then I mentioned already spectral unmixing within Inform. So we need to choose several representative MSI files to generate that unmixing, and then we can perform a batch analysis to unmix the rest of the images from those MSI regions. The final thing we're looking at is data analysis. So there are a large number of different data analysis softwares available. The ones that we use most within our center are Halo, and I mentioned we have 8 licenses of Halo, Inform as well. And there's also other softwares available such as Phenopter, Visiopharm. QuPath is a good one because it's free. There are also a number of R packages which have been developed by researchers here at Peter Mac. So we have ISAT and SPIATS. And at the bottom there, I've put some links if you guys are interested in finding more about those R packages. The workflow for data analysis, as I mentioned already, we're using Inform to perform the spectral unmixing of the raw data. And then within Halo, we're going to fuse all of the individual images to create a fully scanned tissue image. We can then perform tissue segmentation, annotation, and classification on that image. We'll go through cell segmentation, cell phenotyping, before finally doing spatial analysis and then exporting the data out. And again, Meta will go through more in depth each of these steps in her presentation. So with that, that brings me to the end of my part of the workshop. So we've given you a brief introduction into how OPAL multiplex IHC works. We've touched very briefly on immunostaining protocols, image acquisitions, and data analysis. And again, Rayhan and Meta will expand on that within their presentations. I would like to thank all of the members of CAM staff, especially Rayhan and Meta, and a number of the vendors as well for help developing the presentation, and also running some of the equipment within the core, and obviously those foundations which have supported the center in terms of purchasing equipment. Thank you very much, and I'm happy to take any questions.